All right, hi. We're gonna get started. Thank you everyone for coming out on a rainy day. I know it's kind of gloomy out today, but we're really excited for our two speakers today at MIA. The first of which is Hanchen. Hanchen Wang is a joint postdoc at Stanford with Yuri Leskovic, and at Genentech with Aviv Regev. He works on AI for scientific discovery and therapeutics broadly and earned his PhD in computer science at the University of Cambridge with Joan Lazenby. Some of his recent work, which I'm sure he'll touch up on a bit today as well, includes like discrete diffusion, DNA and protein design. And so we're really, really excited to have him here. Both of our speakers did come from California. So if you're doing the math, it's 6 a.m. their time. And so we're really, really appreciative of you guys. Thank you.
Thanks. You can hear me, right? The micro is on. Yeah, I don't need more introduction. So the structure of today's talk will be, as I'm giving the first talk, I will cover like the basics of the agents, like I don't know, like 20, 25 minutes. We'll talk about the agent we built specialized for spatial biology, and we will cover some new recent agents that are doing massive money on aging intervention, as well as using computers to analyze cells, as well as doing a better statistical calibration on the different kinds of agents on different kinds of data sets. Yeah. So if we think of the biomedical discovery, it's roughly can be divided into like 3 stages. You have some observations, can be from some previous data, it can be from the nature, or can be from some literature, and you generate some hypothesis. The hypothesis can be with different forms, like whether there's a direct relationship or causal effects from gene A to a phenotype B, those kinds of formats, and you design some experiments to verify that, to collect more evidence, to collect more data, to help you to refine or reject or accept your hypothesis. So it's roughly, you can think of it as like a 3 interconnected and circulating stages until you find something you wanted or until you run out of your budget. And we see that computation helps at like every single stage of them. And there are some examples, like for instance, you can use some source of autoencoder, some variations measure to decide like which genes or which are more informative for the tissues that you are interested. Or there's like a different ways that can use graph. You can use the relationship of the gene co-expression patterns to help you design, help you predict and design what would be the next rounds of genetic perturbations. And to speak of the observations, I have to say good words to shout loud for the wide collections of different kinds of efforts. For instance, omics data, for instance, other types of data as well. And hypothesis. One of my favorite example is like the double helix structure of DNA. It's back then like 70 years ago, when these two Cambridge alumni, they proposed a hypothesis about the structure of DNA is just based on some pairwise distance metrics from 2D, and it's I would say it's a quite elegant example for hypothesis generations. And there are more examples recently used different kinds of computations. Some are agents that help us to verify or generate some better hypothesis. And the task can go long and long. For every single stages, and there'll be some computation methods that help us to make the process easier, faster, and better. And so what is AI agents? In some of versions of my slides, I usually give a formal definition of the AI agents, but today let's start with an example. So this is kind of the analysis I've done like last week. So when we like back and forth on a manuscript that is about to be pre-printed very soon. So basically we've done a lot of interventions of the mouse models. And so our PI is commented in the document saying that you need to find more evidence in the human data in supporting what you have been found, such as a special type of inflammatory fibroblast or inflammatory macrophages that we know that are critical for the cancer therapeutic reactions. So you need to find some evidence like shared, does the same population happens a lot in the human single cell RNA-seq data as well, or it's have the similar dynamics or similar pathways. So it's kind of like a very high-level instructions, and that's me. I converted it to a few sentences and plug into Cloud Code. So I gave relatively detailed but still very high-level instructions. Ask the Cloud Code to, which is a coding agent you can think of, to create a Jupyter notebook to project the human prostate cancer fibroblast onto the mouse cancer-associated fibroblast, and transfer the subtype labels from the mouse to the human. Also load the, list the data paths for the two data sets. Use the top 2K highly variable genes intersections, so which involve like a conversion and find the intersections of the symbols from the mouse and to the human, and then make some analysis and visualization as well as save the results. So the Cloud Code, it start working. The engine is on and it's generate lots of code, first the general plan, and its final solution, which only involve like 2 or 3 additional inputs from my end. And it could generate a lot of a working solution, which include mapping the mouse and human shared gene space, and project the human data to the mouse, transfer the label, use a conventional method that is shared, used by lots of public literatures, and doing some visualizations, the U maps, the confidence score, the proportion comparisons across different conditions, as well as plot the markers for different subtype limitations, as well as save the results. So this is all figures that are generated by Cloud Code. The top left is a mouse reference data set. You have different types of fibroblast, and you project the human data, which is from the cancer progression samples. You project them onto the same coordinate space as shared by the mouse. And as well, you have the confidence, as well as you have the distributions, which is the top bottom right. And we find the evidence we want, which is this specific type of fibroblast, CCL2 enriched, which we know it's play an inflammatory role, and the larger population is, which means the more therapeutic potentials you will have. So essentially, we find the human has even larger proportion for this specific cell type, which stress the findings we find in the original paper. And this entire workflow, this entire pipeline takes, speak of the API cost, it takes less than $3, and the total include a few back and forth from between me and the Cloud Code, it takes like less than 20 minutes. So this is kind of something that is already functioning and at least make my life easier. And also to use the AI agent to help with better science, specifically on biomedical discovery, is also a vision shared by lots of industry leaders, like Anthropic saying, actually the CEO is also trained by a biologist back then he was a PhD and postdoc. So he's saying the right way to think of AI is to treat it as a virtual biologist who can perform like a co-pilot to all the tasks human scientists do. So not only like a forward pass to do like a protein structure prediction, but also to do a complex chain of actions towards a high level goal, as well as the same vision shared by DeepMind, as you can see from lots of their publications, as well as OpenAI. And a generic structure of the AI agents can be shown as the left paradigm. So it's powered by a large language models, and it can take actions, usually from two callings nowadays, and have some impact on the environment. Environment can be, let's say, your local computation platforms, as well as, I guess, Kexin will show some example like manipulate like robotic arms to do more physical interactions with the world. As well, you can receive some feedbacks, such as from the human, such as from the environment, like whether the code is successful or not, whether your current design is good or not, and it knows when to start as well as when to stop. This is like a quite high level, but I still like this paradigm. Later on, we will introduce a lot of modules, but which will make the sense more complicated. But essentially, you can remember it's powered by large language models. It can take actions to influence the environment and receive the feedback from the environment and adjust itself, as well as know when to stop. Also have the interactions capability with the human. And if we dive deeper on the agent, roughly saying there's like 3 modules inside of the agent. You have tools, which is the major sense to generate actions to impact the environment. And the memory is, and the planning, you can think of both of them as part of the context. So the memory is stored for its goal as well as the progress, and the planning is like make a concrete plan to take actions according to the plan, roughly saying something like that. And there's many, so for every single module, there's many techniques that helps the agent to make better planning, make better memory, as well as make more accurate and concrete actions. So for the first, like, I think we all know, like, but we cannot avoid that. So every, if you look at some public GitHub package, you will see that a lot of prompt instructions nowadays still involve these basic modules. So one of the modules is called chain of thought. It's very simple and it's but it's so powerful and elegant. So basically, what is showing that, so instead of just ask the LM the question, you want to provide an example with a few chain of thought process. For instance, in the example here, instead of doing a single question on calculation, like a 5 plus 6, you want to provide a few short examples for the enhanced learning for the LM to capture, to have a better idea, like what would be the inference process looks like. And another thing, which is, I believe, or shared by all the agents currently, is called REACT. So it stands for reflection and action. So it's also have the similar philosophy as the chain of thought. So instead of provide a sorting, a sync chains, it's provide more like a pattern. So ask the LM to reflect, like what is the current status, what you have, what information do you have, and what's the objective you want to achieve. And then to do the actions and also update its observations. So this is kind of like a very generic principles, and every single agent nowadays you can find they have some shared statistics in their instructions or so-called prompts. And second, next we'll move to the memory. So the way I think memory is like it's a giant context window. It's an intelligent retrieval machine which allows the agent to record the user's behavior as well as the user's information. So this is like one of the example we borrowed from MemGPT. The memory usually have like few functions. For instance, like first it will create memory. For instance, you have some interactions with your chatbot, discuss your birthday, and the chatbot will, if it's a first time conversation you discuss on this topic, the chatbot will create a memory to memorize, okay, today is your birthday, February 7th. And later on, for your follow or for your future conversations, your chatbot or your agent will have the capability to retrieve the memories. It will know you better. It will have a more tailored for your own interactions, as well as the update, like your breakup with your boyfriend or girlfriend. So the memory will also change to update its knowledge. And you can, and the system design actually is not that trivial. So in this paper, is one of the representative paper to build a memory system for the agents. It's essentially have a lot of layers and modules that just help the LM to make a better use given the budget of its context window, as well to update them with few time latency. And you can easily think of how we can use the memory in terms of the biomedical discovery. So it can help to learn the lab's know-hows. I mean, even there's like 100 labs doing perturbation experiments or analysis. There are still different labs who have their different know-hows and preference. So having agents that can memorize the lab know-how, the lab's taste, it will help you build better workflow and analysis, and it help also tackle the hard tasks with long horizons of actions. Because you have a good memory system, you can memorize what's your current step. Because usually there are, for instance, like playing Pokemon or doing large simulations, the runtime of the agents can last for hours or days. So in that way, you want your memory system not only to keep track on everything, but also intelligent and compressed during its progress. So having a very good memory system is also kind of crucial for building an intelligent scientific discovery agents. And you also want to avoid your previous failures or precise failures. Sometimes the large language model will make very silly mistakes. You want to avoid that after you credit like one or few times. So memory would help with that, ideally. And next, we're going to move on to tools. So the tools, I believe, is kind of...
You move on. You're outlining these components. I'm curious in practice whether people do they fine tune the models? You have the prompt, right? But do you also fine tune the model to be a good memory system? And do you fine tune the models to work together, or is it like just everything's better specified through the prompt? And
I actually prefer no fine tune. Many papers they fine tune on, let's say llama, and usually with a fixed context window like 8K or a few thousand K. But if you look at some like a very maintained open source project, like some, let's say it's called open cloud code, but it's kind of like open source solution for cloud code. They essentially write a very detailed prompt or ask the Sonnet or Opus itself to compress the context window compressor map. So it's kind of that side. And so speak of the compression, speak of the tracking a single run, I feel like it's more elegant to just put the case in features. But speak of like, let's say if you have some interaction with a system last year, and you still want to have it to remember such information, like for your today's conversation, I would feel more safe to build a very intelligent retrieval system instead of fine tune a small retriever. Yeah.
I see. So there's some choices there, but I guess beyond the memory, there's also, as you were saying, like the planning stuff, and these components are all interacting. So do people fine tune them to like interact in terms of how well they perform on the overall task, or is it kind of just, okay, let's build the best possible memory system, let's build the best possible planning system, let's build the best possible tool calling system, and then just hope they work together?
I mean, there's basically like two types of fine tune. One is supervised fine tune. Second is reinforcement fine tune, which or like R-based fine tune. Most on both sides, most way I think is more like tool-based fine tune. It's like you want it to make optimal plan. You want it to find the right argument for calling the tools. I didn't see many like a joined tuning include the memory system. Does that answer your
question? Thanks.
Well, I should repeat the question. Never mind. We should repeat the question. Yeah. So the tools is where you can make a lot of efforts because it's kind of the sense the agents will interact or have the impact on the environment. And there's a different categories of tools. For instance, you can have the models or have some bioinformatics tools like Assayverse. And you have database, you can look up database to find the information you need, find the relevant data you need. Softwares can be also treated as tools. So not only coding based, but also computer use, like you have a graphical user interface. You can move the mouse, you can click the button, you can drag the menu and select the things you want and adjust the range using drag or using drag or draw. coding for sure nowadays. I think we all witness like by your own experience with using Cursor or Cloud Code or GitHub Copilot. You will see, you will have a sense like how coding, how good they're good at coding nowadays. And a broader view of tools you can think of, like other agents can be treated as tools through more standardized protocol like MCP. And also like our human scientists can be also like treated as tools to provide additional feedbacks or corrections for the agent's behaviors. So this is like our overview on the modules of agents. You have memory, you have planning, and you can take actions by calling the tools. And this is kind of the shared design for all the works we've been built, like a portable agent, you have actions, also the memory to remember what action you've done. You have a pool of actions, which is a combination of tools and databases, and you have some abbreviations for the impact you made on the data, on the environment, as well as Biomni. I think Kexin will share more on how we build that. We spend tons of time on building the tools. Also, Tool Universe, it's from here, from Marika's lab. So they also push the extreme of a better tool system. Like you want to find the best tools, you want to call them, you want to manage them, you want to retire some of the less frequent or unqualified tools, as well as add new tools. You want to combine them together. You want to optimize it. Let's say with a better arguments or with a better adjust a few lines inside of the tools. And next, we're going to talk about some frontier eye of the agents. I would say it's very parallel, and I just list all the topics that come to my head. So it's not a comprehensive list, but I hope it can give you some overview of what people care about in the recent 3 or 6 months. So first is multi-agent system. I mean, there's already a paper published, let's say from James O lab, which is use a lab of virtual scientists to help us design a better antibody sequence for COVID-19 diagnosis. But still, I think we're still waiting or not satisfying, like what would be the best optimal multi-agent system. So this is kind of one of the diagram from DeepMind's co-scientists. So they spend most of their computation budget on this multi-agent system, which is essentially to create a hypothesis, a useful hypothesis, or useful plan hypothesis. And if you think of like and it's more like a role play. So it's using all the same LM, but with a different instance and with different roles. So you can think of the advantage of the multi-agent system is like it can speed the context, it can speed the tasks. Because sometimes you have a very complex task, it's always easier to accomplish it by divide and conquer. So having more agents that tackle different angles of this problem will help you to achieve the tasks with a higher success rate. And also it can add some diversity. For instance, your agent can have the different access to the tools, your agent have the different expertise. So having them discuss with each other will help some like different angles for the question you care about. And second is self-involved. There should be a video here, which is like a Minecraft, like a character in the Minecraft that is like involved itself. So this is a paper from NVIDIA actually like 2 years ago. Yeah, pre-printed like 2 years ago. And the task is very simple. It's asks the character to learn to use the diamond tools in the Minecraft world. So actually the diamond tools is one of the very high-end tools in the Minecraft. And to achieve using the diamond tools, you need to first to develop the wooden tools, which allow you to mine the stones, and you will have the capability to create the stone tools and the iron tools, and finally diamond tools. And they create a reinforcement learning policy models that allows allows, which is more like embodied agents in that simulation world, to try with different policy to explore and exploit to find the optimal, to find the way to create the diamond tools and have a better survival status. And this is also used by, this kind of philosophy is also shared by lots of recent work like AlphaInvolve and many other works. Next frontier is context engineering. So this is kind of like a hacker word, like especially 2 months ago. So everyone is talking about context engineering back then. And so compare with the traditional like prompt engineering, which is like a single term query, you have the system prompt as well as the user message. Context engineering is more like a more comprehensive engineering. Is, you can say, well, it's a natural progression of prompt engineering. So the reason we need context engineering is like based on two observations. First, we have very limited context windows. Second, we cannot fit all the informations. Second is, even we have infinite budget of the context window, the RM's performance, the agent performance won't be better if we feed all the information. Actually, because the intrinsic computation complexity, the quadratic complexity because of the transformer, especially calculating the cross attention scores, usually is the shorter information, shorter, accurate, and diverse information you provide within the context window, the better the agent will perform. It's like you think of like annotating like a cell type or doing some analysis. It's not always like you provide all the informations, we will have the best annotation. It's more like you provide the most salient, useful, informative information to help the agent perform better for the tasks that you're interested in. And reinforcement learning, it's also a very emerging field, and especially in the past months, or past 1 or 2 months, everyone is talking about agentic RL. Lots of people inventing wheels for training a better agents under different signals. So roughly saying, there's like 2 modules. You have the trainer, which include the agent as well as some framework, as well as base models. And you have an environment. You have a generator, which is generating used for training data. You have an inference agent, which involves like a serving system, as well as the agent, which is you can think of like a large language models, and it will take actions to the environment that you receive signals, and you can update your model from methods like a GRPO, DPO, or different kinds of ways. And even and this is very a complex engineering. And even for a single tiny byte of them, such as to analyze the model's behavior on the different signal, on the different optimization algorithms, is still, there's tons of papers on that. For instance, to analyze how to select the most informative examples, the successful traces as well as the failed traces, to using the same algorithm such as GRPO. There's at least you can find like 5 or 10 papers that are just doing this types of angle analysis. Yeah, and it's also worth mentioning there's 2 types of RL. One is online RL, one is offline RL. And nowadays the community trend is more like on the online RL, which you can learn and interact with the environment as well, update your model, update your policy on the same time. And frontier, another frontier is computer use. Yeah, I feel like it's not playing because I'm not connected to the internet. But essentially, a current trend, one of the current trend on building an agent is ask it to use the computers, ask it to move the mouse, to move the to drag the window. For instance, for like cell profiler, for lots of other software, such as from 10x Genomics, such as you're doing some FASTQ file analysis. Many of such operations are not actually interacted from command lines. You interact them from the graphical interface. So in order to have a larger action space, a capability for the agent, you want it to use the computers. such as like the browsers, which triggered quite some noise yesterday. And for sure, the last piece of the frontier is like to build a better rubric as well as benchmarks. And there are some efforts on building better comparisons of the agents, such as agent arena, as well as to deliver better user satisfied results by running multiple agents at the same time to select the best and return to the users. And the last piece of the basic is some common failures. I think this is something that not many people discussed. I mean, we are all a bit hijacked by the premise of the agents, but we also need to have the sense like what are the common failures when we build the agents. So when we start to build the agents, I say like 1.5 years ago or 2 years ago, we encounter a lot of errors, especially silly errors, such as hallucination on some tools that are not existing, or some arguments that are not defined by the tools. For instance, you import some gene labels in the AlphaFold, which is not expected. And the context overflow back then is on GPT-4. It's very easy to exceed the context window, as well as it has very poor performance to handling some simple errors, such as variable undefined errors. But nowadays, this kind of errors are very less likely happen, thanks to the advancements of building a better large language model from those industry leaders. But we still have a lot of a few other common failures. So first is like stuck in the loop, which represents the agent will repeat the same actions for a few more times, even till the end of your budget, and no finish action. It's kind of similar with stuck in the loop is one of the way to have no finish actions. But there are a few others. It's like the agent will sometimes just happen to stop itself from delivering some final results or delivering some or reach its final budget, which is the number of actions we preset. Still, it's have a lot of hallucinations, especially we move on to very specialized domain of the science, as well as shortcut. For instance, if you wanted to, if you want agents to analyze like 100 gigabytes or even larger files, that has like millions of cells or other types of information, it's tend to find a shortcut, which is to subsample data or doing a lot of simulations instead of to work on the actual data, as well as trivial findings. It's like a lot of things we find in today's agents is like either they do a lot of hallucination or find some shortcut, which is something very unrealistic, or they provide something trivial findings, as we all know, like a fibroblasts exist in tumor tissues, those kind of trivial findings. So this is something still exists, but despite of these common failures, we already observed there's many positive signals in building agents. So one of the example I'm going to share today is we built for spatial biology. So the reason we chose spatial biology because it's a very broad, very vertical field, but it have high impact. For instance, one of the example is like to distinguish cold and hot tumors. So the way we design a spatial agent is like as we shown before, is like it has 3 modules. You take actions to have impact to the environment. You have the memory to track your progress, and you can make plans. And we choose representative works from every single stages of the biomedical discovery in the spatial context to support multiple organs as well as cross species. Speak of the time, I will skip the results. It's in the paper is like 100 or so pages of manuscript. So one of the things we're going to highlight is that I'm especially good at aggregating information, like multiple layers of information. So here we ask the LM to look up different database and select the most informative genes, given it's a few principles, such as if the gene used to name the cell type, which means it's quite important. If the gene is included in your target tissue here is the specific region in your brain, then it's kind of very important. And if it's appeared multiple times, then it's important. So I'm doing a very good job in aggregating different types of information resource and ranking them as well as provide reason in natural languages that are very easy for us to identify, to justify. Speaking of performance, we compare mostly with the specialized tools that people are built in this specific task. It's better consistently across different metrics. We also ask a lot of human scientists to do the same task using the same informations. And so first observation we made is like scientists, they are so different. So we ask like 8 or 10 scientists, we summarize their design, panel design workflow, and they're quite different. For instance, some scientists will just use some existing templates that he or she has in hand. Or some scientists, they will do a very detailed check. They will look multiple data sets, many literatures, as well as some database for pathway and markers to find, to adjust his or her design each one by one. And some just use some literature as well as reference data sets. And so what do we show? We compare with spatial agent and human scientists. We basically can see that it's performed the most of the scientists, the data we collected, which are mostly PhD postdoc as well as full-time scientists. And it's kind of proved like 85% of the human initial designs and also agent plus human. Agent work on the human initial design is also better than the agent alone. So this is what we found using GPT-4o 9 months ago. Things might be different if we switch some more than LLMs, which we will have in the revised manuscript. And speak of the timeline calls, it's much cheaper compared with the methods that I use GPUs. And also for sure, you can imagine it's cheaper for the scientists because we know the scientist time is very precious. Annotations have the similar flow as well as similar performance. It has better than cell typist, some GPT-based annotations, as well as most of the human scientists. And we use it to doing mostly to analyze how the cell interact with each other under different contexts, which here is a tissue remodeling process. And as you can imagine, lots of omics-based analysis is on comparative analysis. It's like you have different conditions, you have different ways of grouping them, and you would just want to group them and to analyze what changes and what are the intrinsic reasons. And LLM are quite good at doing this very standardized or pattern finding types of analysis. And there are some similar works that I published preprinted after our work, like Cell Voyager, like Pivotal Code. Both are from James Stowe's lab. And we do some lab validations, which is another manuscript that we hope we can hope we can share before the end of this year, because there's a lot of screening policies we need to make. And one thing I want to say, as a time we build spatial agent, we already consider its extendabilities. So it's like the user make a message, and the agent will decide whether the user's task has been encountered, has been like baked in its previous like plain templates or know-how or SOP. It's different terms, but for the same concept. And if the plane exists, and the agent will make the actions much easier using the template. If it's not, the agent will make the plan on the fly, use the tools it has. And this is kind of the stages we used like 9 months ago. And nowadays, there's like a much more comprehensive modules for this. For instance, like a cloud skills. It's kind of like you provide a repo or provide a folder, and the cloud can learn learn the know-how, learn the preference, learn how you do the analysis, how you make the figures from the examples you provided, and update the memory to update its plan in order to make a better tailored user quests. And we show that, okay, spatial agent, thanks to the coding flexibility, it can do simple tasks, such as harmonizing annotations between two different data sets. But it's still failed a complex task, mostly because we didn't grill its coding capability, as well as we didn't add the tools that are needed. So next, finally, I'm going to talk some recent works we're about to release very soon. I'm so excited for them. There's a few works, so each of them will be covered by a few slides. So first is like in collaboration with here, Vadian School, as well as Tony's group at Stanford. So we basically didn't use a very complex agent. We basically use a coding agent to do a massive mining the aging interventions using the clock system from omics data. So the motivation we need agentic system instead of like a predefined like a Python program is because every single GO study, they're different. They're in different formats to record the metadata, so they're so messy. And you might want to try different statistical tasks based on your grouping, based on your sample size, and you want to aggregate multiple information, not only on the statistical measure like p-values, but also on how the experiment is designed, what are the relevant literatures. So you kind of want to have this kind of flexibilities. Also because of the intrinsic complexity of the data, you want to have a more dynamic or flexible system to write code for each single GO data set to do some analysis. And what we find is this is, as you all know, this is a typical volcano plot, and we show, so the color represents how the different types of interventions affects the aging clock changes. And we find that one of the interventions, which is open, which we verified in the wet lab, we show that it's actually could increase the lifespan of the mouse model, and the phenotype is basically it's helped the hair to regenerate or regrow from the mouse models. So the example here is basically I want to say, in order to make the use of agent, you don't need to build a very comprehensive ones. You just to find what you need, and you can easily use some very simple agentic structure to help you to meet your need. Second project is like we're building playing some statistical games. So we use e-statistics, which is a method actually used in Popper from Kexin and many others, which is like a way to model the sequential statistics, the sequential decision makings. So this is like one of the examples, because as we said, agents still make a lot of silly errors, such as you want to find some loci on the chromatin 17 associated with type 2 diabetes. The agent might sometimes in the middle, it's fine. It uses a different chromosome instead of the 17th. So in that way, you want to judge, you want to verify to identify that, and it has some additional advantages, such as you want to early terminate your agent and start a new run, or you want to save your budget, or you want to provide a measure that the current agent running is not ideal, most likely to provide a failed results. So we use we call our method called e-evaluator, which is like the sequential estimation on how the agent will successfully achieve its tasks. And we show that we'll find with a few serial proving showing that the method we design will have controllable error rates as well as optimal power. And we show that compare with raw LM charge, we compare with some Foroni test, as well as some empirical baselines. And we show that our method is can achieve like an ideal empirical type 1 error estimation, as well as to have a better power, which means you'd require less samples to do robust estimations. And we show that it can generalize across different data sets for different agentic architectures. So here is like aviary from Future House and GSM-AK, which is a mass data set. And this OctoTools, another one from James Low Lab, and on the medical data sets, we show that the same, our proposed method has the same good performance compared with all the baselines. And the last project I want to share, very briefly, is I mean, is an agent that can not only coding but also use computers. So we're still deciding the name. It's whether called Coco or Coca. But essentially, you get a sense of it. So it's a multi-agent system, and you have two agents. One is just doing manipulation, doing operations on the graphical user interface. You can have a lot of interactions with, let's say, cell profilers or QPath or Napari or different types of softwares that work on, let's say, cell images. And another is like a software engineering agent that can mostly run a lot of code and can robust recover from different error messages, as well as for long horizon tasks. And these two agents can communicate with each other and collaborate to achieve a very complex task, especially complex if we think of another layer of modality for cell images. And we compare some baselines. This baseline, so for this type of, so the right is like a two example. One is generated from the agent, one is generated from the tutorial, one is downloaded from the tutorial on cell profiler, which we treated as a ground truth. And we show that this complex task, it cannot be achieved by a single LLM because it doesn't have access to use cell profiler to doing some data analysis on your imaging. And we compare with a few baselines. We encrypt the Biomni because it's originally designed for coding, but I know so we compare with the coding version for Biomni equipped with the cell profiler. We also doing two types of version of our own method. One is just on coding, one is use both coding as well as the computers. And we show that it has a very better performance than most of the methods. Not only on the in-domain test, which is like a trained test split on the cell profiler tutorial, which are like 20 different samples data as well as different tasks, but also for the out-of-domain test, which is we download the most hard challenges from some online forums that is not encountered or included in the cell profiler official tutorial. And many of them have open solutions. And we also analyze when the computer use is necessary. So essentially, you can see that our system essentially use tons of computer use softwares, such as to validate, to verify your results, as well as to adjust the parameters, such as the ranging, the threshold to color your cells, to dye your cells, as well as to just build from scratch, which is like move the button from the beginning to drag the data file into the cell profile window and drag the menu to find the corresponding functions and doing some analysis. So to recap, so we share the vision. Many of you shared as well, like the agent as a scientist co-pilot. And we talk about the basics of agents. We talk about its modules. And we talk about the frontiers topics as well. We share some common failures while we build these agents. And we introduce some specialized agent we built for spatial biology. We built it in the light of to compare most of the specialized domain tools. We didn't compare with other agents, but more we want to show the agents can offer some performance gains across some like a previous computation method, like whether it's a transformer or encoder or some statistical based methods. And we have some manuscript to do lots of wet lab verifications and share some works that will soon be released massive mining agent interventions, validations using sequential hypothesis testing framework, as well as doing coding and computer use at the same time. So finally, I want to thank my mentors. This is because there are so many authors. I kind of lazy didn't list all the people's. Well, essentially, this is one of the slides I borrowed from one of my previous presentations. I want to thank my mentors and the core team spatial agent, as well as predictable agent, Biomni, many agents and many works, mentors and mentees. And thanks for listening.
People have questions, let me know.
Hi, I was wondering if you think that, for example, you could have an agent, but without the LLM component, because I know that the LLM component is providing a lot of the underlying reasoning for a lot of these agentic workflows, but it's also providing the knowledge source as well, where like, if you ask it to run a single cell pipeline, the agent probably is drawing from a lot of that pre-trained data that's been mined from the literature. So I was just wondering how you differentiate, agents as a whole versus the LLM field as a whole, or there sounds like they're inherently intertwined.
Yeah, actually, so as you know, the agent concept is developed better or proposed earlier than the LLM. For instance, in this paper, they didn't use large language model. They use some visual models. For instance, on the very early stages, like from DeepMind, they developed some framework called hide and seek. So basically, that in that way, it's not agent, more as in that it's not LM power agent, more used to analyze the reinforcement learning, how the emergent behavior happens in the multi reinforcement learning policy powered agents. I think for now, it's like for sure, mostly agent we're talking about today or talk about by the community is powered by large language models, and it's avoidable that it has some bias inherited from its trained data. So first, it's getting better and better for base models. There are some people are showing that for some data set like GPQA or graduate level science, the agent already have better performance than the average PhD. Second, it's our human self is also have bias, right? I mean, you can read 10 different cell atlas papers. They have their own bias to do the analysis, to do the figures, to tell the story as well. So one of the way to alleviate the augmented bias from the LM and to bake your own bias, we think is like, for instance, like I think cloud skills or some memories or some like more better context engineering could help that. Yeah, I'm not sure does it answer your question.
Very nice work. I have a question just out of curiosity. Do you guys observe any two usage preference for the agents? For example, you have multiple tools doing the same thing. Does the agent prefer to use one over the other? I would assume like most humans do have their own preference, but not about agents.
Yeah, so we do have, so like I would say Cloud 3.7, start from Cloud 3.7, it's tend to use coding itself instead of to calling like a lot of predefined tools. I feel like it's more like from the training side. It's like I feel like it's the training of 3.7 for 4.5 Sonnet is compared with the product of Cloud Code. So they want to train the model in order to have a very good coding capabilities. So instead of from the tools we predefine, such as some like ScanPy or some other different types of tools. And the question for that, for your specific question, I feel like this is something we realized, but I didn't find an elegant solution. It's like for a single task, you can have several ways. You can use ScanPy, you can use Seurat, you can use different type of things to do saying like a differential expression. Just that's a differential expression, a simple example. For different data, you might want to use the different, for sure, parameters for the same package, as well as different packages. In building of this, I'm not sure about the situation of the two universe, but in building of our agent, we're trying to kind of a bit centralize the tools. We try to avoid duplication of the tools for the same purpose. But ultimately, I think it would be better if someone or from our side, we can propose something that the agent will try a lot of different solutions, like different tools, different arguments, and propose, okay, this one is the optimal in my hand. Currently, it's more like a one-time pass. Even if you look at it's kind of like it makes a plan at the beginning and just make actions according to the plan, maybe adjust the plan in the middle, but essentially, as long as it has a final result, then it's stopped. It won't, okay, I have one version of the result, then I start from the beginning. Currently, we don't have such thing. But I believe have such kind of thing will make a better deliverable things.
Thanks.
For people who have done a bunch of computation in their life but haven't really jumped into this yet, do you have a quick recommendation of where you would go to try building out one of these? Let's say you had a computational task, a machine learning algorithm you were trying to build or something like that. This world of there's lots of choices out there at this point. Can you offer some recommendation as to what to try first and why? Or do you need more specifics on the problem? Let's say it's a pure data science problem. It's a pure, there's a lot of data and but there's a lot of pieces you need to connect together to get it to work.
If we think of like a resource, like something to read, I would have a very heavy bias to read something from like Anthropic. I really enjoy reading their blogs, especially engineering blogs. Reading, if you start from something reading to understand the basic concepts, I would start from reading blogs from Anthropic. They are very aggressive in pushing the agentic power for large language models. If you think of a very good open source packages, I would recommend Biomni and Biomni, which Kexin will introduce in the next talk, as well as some existing a project like a Sui agent, which is push the boundary of the coding capability of the agents. For your own need, you might want to first figure out what kind of the tools or functions you want to have, and you'll find something that is very easy to extendable, such as Biomni to add them. I think personally now is we spend our time is saved a lot by using a coding assistants such as Cursor and Cloud Code, and you could also you find some agentic framework you want to work with, and you download some tools or your local tools you want to use, and you can just translate the tools that are be callable by your agentic framework. Just to use a Cursor or Cloud Code, provided it's a few example and some instructions, and it can do a quite decent job with a few rounds of interactions.
I'm going to move on to the next speaker just for the sake of time, but they will be sticking around afterwards if you have more burning questions for Hanchen. Thank you so much. Thank you so much for being here.
Yeah.
Kexin recently defended his PhD at Stanford in computer science, also with Yuri Leskovic's group. His work also broadly focuses on AI and biomedicine, mainly looking at multimodal modeling, therapeutics development, GNNs, agentic systems for scientific hypothesis testing, which I'm sure we'll hear a lot more about today. He's a Stanford BioX SIGF fellow, and recent projects include what we just mentioned of Biomni, an agent for biomedical sciences, and we're really excited to hear more about it. Thank you.
Great. Thank you. Can you guys hear me? Okay. Cool. I'm really excited to be here and share about some of our recent works. The title is Building a Virtual AI Biomedical Scientist. So I will probably give a really, really quick introduction because Hanchen has already covered a lot of the motivation of why we're building a kind of a virtual AI biomedical scientist. And then I'll delve into our methods and also show some recent work. So as we probably all know, biomedical discovery, super fundamental, impact all kinds of different industries, ranging from drug discovery, diagnosis, even agriculture and so on. But if we look at the day-to-day life of a biologist, they're actually, a lot of times, they're not doing science. They're doing like this manual, complex tasks, right? Because real-world discovery is like extremely slow, messy, and complex. For some examples, like studying some papers on some databases, actually talking to some colleagues, no one knows everything. If you're a web lab scientist, if you want some data analysis, you need to engage with some bioinformatician or computer biologist and wait for a few days and come back with the result. And then design some molecule with some legacy tools, draft protocols, order materials, and so on and so on. In the end, you get some potentially useful sub-results. And the best case, if it takes months, if not years, to get it done. So this basically motivates us to say, okay, part of the reason biomedical progress is limited by lack of crazy innovative ideas. Another reason is just limited by human bandwidth. There's just so much data generated because of the new sequencing technology out there. When we talk to a lot of labs, PIs, they just complain, right? They have 80% data that just sit there. No one's analyzing it. And then there's also many scientists, they have tons of innovative ideas. Because of the limits of bandwidth, they can only do like test one idea at a time. So this gradually means that as the time goes by, the supply of these biomedical expertise just grows very, very linearly, bounded by the human. But the demand of it just grows exponentially. So there's a huge gap. And the gap is basically the missing insights, the missing discoveries. So in order to close this gap, we need a new way of scaling this kind of expertise. And we're able to see that in AI agents, in other domains, it's able to scale this expertise. For example, in coding, the cursor, right? And in the legal industry, there's Harvey. And the agent, Hanchen, already gave a really great in-depth introductions. I don't need to delve into it. But we're thinking, can we extend this AI agent to biomedicine? And we do believe that future biology is agentic. We want to create a virtual AI biologist, automate biomedical research. And there's a lot of benefits that it can bring, right? Because for example, like now it can perform tasks much, much faster. Well, we have seen many, many examples where it took months of work, now it takes minutes. And also it enables tackling many tasks at the same time because everything happens in silico. So you can actually have 20,000 agents working for you at the same time if you want. So this actually remove a class of problem that we met before. For example, because you can only do one gene at a time, you have the gene selection problem. But now if you have 20,000 genes, then you can just have 20,000 agents looking at one gene for each. Right? So there's a lot of problem can be solved with agent. That's kind of impossible before. And also, of course, because of the reasoning ability, the intelligence layer can also suggest and explore novel hypotheses as well. So we think this is a new way of doing biomedical research. So in the future, it's hard to imagine there's still people spending all this manual complex work. It's like this kind of artisan era in contrast to here right now. In the future, biologists will be working with a team of AI agents, just get all the tasks done. And the impact is like a single biologist now can have equal to impact of the entire specialist team. Like geneticists can actually now design proteins. Web lab scientists can now do all the bioinformatics tasks and so on. And also this is a great democratization tool as well, like great education tool. Because now if we can encode all the knowledges from here and then into the agent and then distribute it to anyone on Earth, then we're dramatically reducing the barrier and everyone has the same educations. And also, it's also the first time where every step of the discovery process are being documented because agent does everything. Right? So you have the very granular step of, okay, you're using this tool and then that tool. So it becoming a record. So it's also becoming a system record that can of the discovery process itself. This also has a lot of a lot of benefits as well. So overall, we think this is a kind of new way of doing biological research. So this talk, we're basically trying to motivate, this is great, but also, I guess, dive into more technical side, which is how do we build such an AI biologist? So to build such an AI biologist, we also need to look at how does biology research being done today. So here I'm giving an extremely high level, some kind of overview. So broadly, you will need to generate a hypothesis. For example, this is a hypothesis. And then usually how you generate a hypothesis is reasoning, through reading some papers, also doing some kind of analysis as well. And then given a hypothesis, you need to validate it. So typically, for example, a biostatistician could look at these multi-modal datasets and then write some simple script to generate p-values. Or it could be a more targeted wet experiment, for example, knocking out the gene and see what's the phenotype changes. But overall, you're able to see that generating hypothesis and validating hypothesis, it involves multiple individual tasks, and then to do this kind of a more high level kind of a task. So if we summarize in a nutshell, we have the outer loop, which is the hypothesis generation and the validation loop. And once it's validated, you can you basically add it to your own knowledge, and then you can generate more hypotheses. So there's the outer loop, and within the outer loop, each step there's massive number of individual tasks and the workflows. So if we model the biomedical research in this way, then it basically naturally gives us a recipe for building an AI biologist. Basically, we need a generalist biomedical agent that can perform all kinds of different individual tasks. And then we also have a genetic system where we can just compose everything together and then to do the generation and validation of hypotheses. So this talk, I'm going to focus on the foundation layer, which is the generalist biomedical agent, which is called Biomni. But we also have some work, which I will briefly, in 2 slides, talk about as well in generation and validation as well. But I'm going to spend the most time talking about the Biomni project. So yeah, so I guess I want to first acknowledge my co-authors. Biomni is a big project, collaborating with amazing people, Serena, Hanchen here, Jerry, and Minta, and also with our collaborators and advisors as well. So now we're looking at each individual tasks. Now, if we zoom in, if you look at how is the task get done, it's usually a loop of reasoning and actions. So you reason about it, and then you think about it, and then you perform some stuff, and then you look at the result from that kind of actions, and then it will inform you the next step, and then just continues. So it's this kind of a loop of reasoning and actions. And we know recently there's tons of progress in reasoning, but actually the biomedical action space is like undefined. So what do we mean? Basically, the LLM is like it's kind of disconnected to all the things that a biologist have access to, right? So the machine learning predictors, wild experiments, database, bioinformatics tools, so on and so on. And also, so basically the challenge is that we don't have an environment. It's almost like a scientist just can keep thinking without doing anything. Just then you cannot do like real discoveries. So our motivation is, okay, so there's no environment, let's just build one. Then the first challenge is like biomedical action space is super, super large, complex, and scattered. So we need a method to try to create an action space at the first place. So we create a new method. We look at bioarchive, because in bioarchive there's like 25 subjects, and it covers the biology very broadly. And then in each subject of the bioarchive, we retrieve 100 recent published papers. And for each paper, we ask another agent, which is called our action discovery agent, read through each chunk of paper and ask the agent, okay, what kind of tool, what kind of software, what kind of databases that are necessary to reproduce the result in that chunk of the paper. So after screening it for like 2,500 papers, you get a collection of tool software and databases. And if you think about, theoretically speaking, if you implemented everything really, really correctly, then you can kind of reproduce all of these recent published papers in an automatic fashion, given the reasoning abilities is really good. So, but of course, that's theoretically speaking. But I think after this step, you'll get a good snapshot of the possible action space out there. This is our kind of a North Star, but it's actually not implemented yet. So then we go ahead and then try to implement some of it. But because there are just thousands of tools and software and databases and super scattered, so for the first version, we just picked the most popular and the most frequent subset of it. And we initially also tried to let the LLM to create its own, just have like an action generation agent as well. It just does not perform really well. So here we curate like a manually kind of a very high quality subset of it, which is called the Biomni E1. E stands for environment. So in this environment, we decompose it into 3 parts. The first part is the softwares. So these include R packages like D62. There's also Python packages like BioPython, IKImage, and there's also command line tools like GCTA, P-Link, Muscle, and so on. So here we're not defining predefined functions, but we just create a single like software environment that's compatible with all of these. And then the LLM, because it has read all the documentation of these popular softwares, it can automatically generate a code to get a task done. And this is much more flexible compared to you predefine some functions based on these softwares. And then there's the databases. So for these databases, it includes like Nomad, Open Target Genetics, BindDB, Cell by Gene, RegulonDB, ClinVar, and so on and so on. There's 59 of these, and it consists of two parts as well. The first part is this more massive web databases, like terabytes of data. So for that, you can also define a large number of like predefined functions to cover this ID to that ID, for example. But we think if I want to be general, we're actually using a sub-agent that takes in a natural language query and have another LLM to generate the SQL and the kind of GraphQL queries to extract the relevant data sets on the fly. So this way, it's much more flexible. And of course, that's the large web databases. And there's also the data lakes where these are like private, like a list of files where there's no API endpoint, but it's just like raw data sets with like pandas, data frames, and so on. So the software and databases are the core, but we also know kind of LLM actually sometimes cannot generate on its own because there's so much private knowledge in biomedicine, so much know-how. And also there's also involved tools that are really complex. So here we include only tools that are very hard for LLM to generate on its own. So these are the specialist tools. That's how we define it. So this include, like, for example, you want to predict ADAMAT property, you need some proprietary models. If you are doing some WiLab cloning protocols, so we need to like manually talk to some lab to grab some protocols from them. If you are looking for some AI models like UCE, IFDOC, so these were tools that we give the LLM access to in our first environment. Cool. So that's the Biomni U1. And next, we also build an agent that kind of customized for this environment because this is the first environment that's really massive, huge compared to the other like environment, like for example, the coding agent environment, it's like 20 tools and a very simple software environment. So in order to accommodate this new environment, we developed some customized modules. So first of all, for example, retrieval, because you cannot put everything into the context of the LLM, like it's just so much of them. And also most of them are actually not relevant for the question at hand. So for this single, given a question, we have a retrieval step, which basically retrieve the most relevant subset of tools, software, and databases that are needed to answer this question. And for the retrieval, we also experiment on different kind of techniques. And in the end, we realized that actually retrieval also requires reasoning ability as well. So we put another sub-agent that does the retrieval as well. So in the end, you will get a subset of relevant resources. It's almost like a sub-environment just designed for this task. And after you retrieve it, then you can do reasoning about it. You need to think, okay, what's the steps needed to utilize these resources and also the reasoning abilities to answer the question at hand. So here in Biomni, we kind of prompted to create a plan on the fly. So part of the reason is like we want to be a general purpose AI agent. We don't want to be like a very, very focused agent. Part of the reason is like we know in the day-to-day life of a scientist, there's just so much tasks, so much diverse tasks. If you are using like each point solutions, then you're still kind of need to switching different tools. It's complicated. But if it's like a single general purpose agent that you can tap in any question and it give you some answers, just much more convenient. But in order to do that, then you cannot like predefine workflows or like predefine like a concrete steps for every task out there because there's so much tasks, right? That's part of the reason we want the LLM to generate on fly the plans to tackle any given questions. And we're pretty surprised to find that LLM has read all the papers and all the literatures and all the internet. So it's able to generate a pretty reasonable plan. And of course, there's some very, very specialist tasks. And we have some other approach trying to tackle that. But for the most common popular tasks, we're able to observe that the plan generated by the LLM is actually pretty reasonable to give you an answer. And also the plan is actually tracked along the entire process. Maybe because maybe sometimes in the middle, there's some wrong answers, and then you need to update the plan. It will also automatically up the plan as well. Cool. So now you have a plan. Now you will need to really get a task done. Right? You will need to perform the steps. So for each step of plan, we use coding as a way to solve to kind of perform the step. So I think this kind of already become almost like a standard, as Hanchen mentioned, with the call code and things like that. Like when we develop it, actually, it's actually like 1.5 years ago. So it was still a bit unclear, like coding is the way to do. But for us, we are using coding because it's almost like a necessity. Because for our environment, we don't define any pre-specified functions on the softwares. So it has to use coding to execute these softwares. And also we wanted to interleave the software databases and pre-specified tool intelligently. So coding is basically a way of general task solving that can interleave them and also can do like more complex logics. For example, for loop, if and else. This just gives much more flexibility compared to this kind of a direct function calling approach, where it's constrained by the list of tools available. So this is a kind of general overview of our agent. It's actually very simple, it's not like a very complex architecture. Part of the reason we also, as I'll also mention later, we're going to train the models as well. So having a simple agent architecture also gives it's much easier when we do the end-to-end kind of RL agent as well. So just give an overview, give a task, give a question from the users, and you have a retrieve from the relevant tools, and you will reason about it, and then you will code, and then you have an execution result, and do more reasoning and more tool use and so on. And until you think, okay, this is the answer that can return to the user. So that's our method. And then I'll delve into tons of evaluation that we did to show that it's actually have 30 signals. So we first utilize Fusion House Lab Bench. So we use the development set from like 45 questions from these datasets just to tune the agent, just tune the tools. And these are like database queries and the sequence manipulations as well. And on holdout test set, we're able to see the Biomni achieve human level performance on these two specific tasks. And it also outperforms all the baselines, including LMs, React, coding agent, literature agent, coding plus research agent. And also, this is the ablation, where we, instead of using coding, where we use the tool use. But this is kind of more like a sanity check, because we have the development set, right? So here, on completely no development set benchmarks on humanity last exam, our agent is also able to perform pretty well on a completely zero-shot settings. But actually, all of these three benchmarks are like exam style, like multiple choice questions. They are not like real world research questions. So we set out to kind of go ahead and then create a new class of evals that correspond to real world research. So this includes like GWAS causal detections. For example, you have GWAS hit. There's a neighborhood, there's many genes. Which one is the causal genes? And then the perturbation screen design, you want to design a screen. You want to select 50 genes for like T-cell exhaustion, which 50 genes to select. And also single cell annotations, microbiome disease text analysis, repurposing, rare disease diagnosis, and so on and so on. So these are snapshots of the thousands of tasks out there, but it's a snapshot of some of the popular tasks out there. And for all these tasks, we're able to get some proxy of the ground truth from the users. For example, the collagen detection, we have some expert annotated deeply. For the perturbation screen design, we have like for this specific cell, like context of interest, some people have already done the genome screen. So we know whatever 50 genes to choose, we already know the post-perturb effect. And for single cell RNA-seq annotation, we have the author annotation as well. So these are all like some proxies of the ground truth, and we can measure how good is the agent aligned with this kind of proxy. And overall, we're able to find that also completely zero shot, no development set, our agent is able to have the best performance for that. But still, we feel like this is still also some benchmark with some kind of proxy of the ground truth. I think we really want to give this tool to the scientists, let them try it out and then see if it's really kind of useful for them. So we first partner with a few labs just to test it out. So for example, we work with Le Cong's lab. So Le was doing a lot of functional screening on like CRISPR's gene editing experiments and so on. So one of his students was doing this cloning. So I have a plasmid sequence here. I have to clone a CRISPR guide RNA targeting the B2M gene into this plasmid. I want to generate a plasmid map and a step-by-step guidance So instead of human do it, it just give the task to the Biomni, and the Biomni just go ahead and then do the plasmid analysis, guide design, oligo annealing, and so on, so on, so on. It takes around roughly 8 minutes to complete the task. And then Biomni is also able to generate a web app cloning protocols in details. For example, the oligo sequence here, the primer sequence here, and also each step-by-step plans. And the scientists basically need to follow the protocols and then perform the cloning experiments. And Jerry, they're here just really perform the experiment following the protocol and is able to find this successful. And he's happy. So I mean, cloning is not like it's not the craziest novel kind of task, but it's like super frequent, super kind of involved. It still takes some time, right? A few hours, and navigating from different tools, snapgene, AdaGene, to generate one construct. But Biomedical does it only in 10 minutes, and they just need a natural language prompt and just wait there. And after a coffee, you will just get the result. So later we kind of further want to test it out to like more rigorous settings. So Jerry here created 10 more tasks that correspond to his like day-to-day gene editing experiments. And then we recruited a trainee with a Stanford master students, and she has done cloning before. And also recruited an expert who's a postdoc in Le's lab, and she has done cloning for the past 8 years, so like a real world kind of an expert. And we asked, I also do the LLM and Biomni, and for all these 4 entities, we asked them to do this 10 tasks. And then we have another expert just blindly review all these 10 tasks and rate how complete it is, how accurate it is. And then we're able to find that Biomni has pretty similar grade as the expert, and it's much, much better than trainees. Of course, much, much better than LLM. LLM just kind of hallucinates all the time for these kind of very granular protocols. So actually, this gap is actually really interesting because the trainee, like usually, trainee needs to bug the PhD, the senior PhD student to get a task done. But now actually, you could just need to talk to Biomni. It has similar performance. Biomni will never say no to any request. Yeah. So that's on the web lab part. And then we're also able to find that the agent is really, really useful for the dry lab data analysis as well. So here we partner with Microstandards Lab with Minta. Basically, they have a lot of wearable sensor data, like glucose monitoring activities, and they're like 458 raw Excel sheets, like super, like there's a lot of missing data in there. It's just a pretty manual and kind of complex task for humans. But the more complex it is, actually, the more suitable is the agent, right? Because the agent just feeds into the folders, and then it just goes in and then just iterate over everything. So we give the task to the agent. It have the dataset preprocessing, doing all of these steps, and after 35 minutes, it generates tons of visualizations and also generates some interesting conclusions. And interestingly, Minta, when she did the same analysis, she also has a similar kind of conclusion. But the agent actually did a much more visualization, explore much more experiments as well. And it took her like 3 weeks, and working 2 to 3 hours per day to get a task done. But the agent was able to complete it in approximately 35 minutes. So we are able to see that on this kind of manual complex data analysis, agent is tremendously helpful. But actually, for this example, it's all like Excel sheets. So all this kind of it does not require like specialized software packages to do some processing. So here we are actually also demonstrated with a more complex example on the single cell RNA and single cell taxi data set, this multi-omics data set, where it involves very complex softwares and a choice of databases and kind of the steps. So Hanchen here was wanting to analyze this multi-omics data sets, and then it gives some kind of detailed instructions, and the agent just be able to follow the instruction, use PySync, Jiang and Boost, this target, and so on, so on. And after a few hours, it generated all the visualizations and also generate kind of some interesting findings as well. So here is this novel TFs that show hierarchical activities across the skeletal lineages, which is not reported in the original authors' papers. And also the Jintian and Peng here, who are the biologists also manually check the traces and find that they actually align with how they also perform the task as well. So cool. So these are some like quick case studies that we demonstrated in papers. And since the launch of Biomni, we also increasingly realize that we want to basically create an operating system of biomedical research that just can defragment all these database software tools and putting them into the same roof. We make the open source package like really, really easy to integrate these new integrations. And we also realize that every new integration enables new task capabilities and a compounding way, right? Because you add a new tool, but this tool can be used by multiple tasks. And then keep adding this tool, you're unlocking more and more kind of capabilities. So here, just want to demonstrate a few of these examples. So we partner with SDVerse, who kind of host various lists of softwares for single-cell spatial and perturbation data analysis, such as PertPy, ScanPy, and like SDVI and things like that. So here, after adding this software to the Biomni, it's able to do like spatial data analysis, for example. So here we give an example, like given a sick fish data set, we ask it to explore it freely and analyze it and identify interesting structures and generate some hypothesis. So it made a plan, and then after a few hours, it's able to generate a few interesting hypotheses, and also with the proposed experiments and the expected outcome as well. So that's on the single cell analysis, and then we also partner with Tamron Bio, who's like API providers for these protein design models. So here we can now do protein design with Biomni. For example, given the starting protein sequence, you want to iteratively optimize its thermal stability. So Biomni, basically, given the sequence, it kind of make the plan to use AlphaFold to make a prediction on the protein sequence, and then use another model, thermal MPN, to predict the thermal stability. And then it look at different databases, literatures, and also look at the predictions and the structure itself, and propose the mutations to modify the protein sequence to optimize the thermal stability. And it does it in like several rounds until it kind of improves on the thermal stability. And so here's the answer, like all autonomously. It's like this is the optimized sequence, and this is in each cycle what is the mutation proposed, and this is also the rationale. Right here, this at the early 83rd positions, the transfer from like Q mutated to I, and the reason is like it has enhanced hydrophobic co-packing and so on. So, and it's also able to see that the thermal stability also just kind of increases as the cycle goes by. And also we partner with PyLabRobot, which is like a Python interface for all kinds of different wet lab equipments like Hamilton, OpenTron, and so on. Because we think in the future, if we really want to build an AI biologist that can automate everything, like from data analysis all the way to wet lab experiment, all the way just to give you something, like a very long horizon fashion, the wet lab part is definitely a big bottleneck. And I think it's still very early right now, but we partnering with PyLabRobot to demonstrate some early capabilities. So for example, right now, you can just use natural language queries to orchestrate like liquid handlers or some other robotics as well. So here is a demo. So this is really Biomni in action. You just put it in a natural language query, and it's able to basically move kind of plates from plates. And in the future, as PyLabRobot is kind of expanding to more and more robotics, covering kind of more operations, we're kind of thinking like, in the future, it's like another agent create a prompt for the PyLabRobot to do it, and then it will also return the result, and then it will inform it to do the next step, and it just continues until it has the answers. Yeah, as you can see, it successfully moved from wheel to wheel. Cool. So these are also just kind of some demonstration, because it's a general purpose agent. It can do all kinds of different capabilities. So we're able to try a few things out. Like, for example, you can do variant interpretations for human geneticists, and we can do automatic profiling for drug developers. And it could also do some like manual tasks, like download some random data sets online, which actually takes some time as well. And then also do like screen designs, as I mentioned, also do some real disease diagnosis, because often it involves more than just the patient phenotypes, but also you give like the VCF files, then you will need to basically run some bioinformatics pipelines and using reasoning ability to integrate it with the phenotypes and then create a diagnosis. And also docking, sync annotation, and so on. Yeah, so we open source Biomni, to public, and since then, I think a lot of people are contributing new tool, dataset, and softwares as well. And also we also add like MCP support, so we can expose it to the like other like cloud code and things like that as well. So we also build a web platform that for scientists that can quickly just give a natural language query and then it will get the task done. So here, this is the platform interface. So I'm just giving you an example here. Here, for example, this is the spatial data analysis example. Here, initially it makes the plan, and then you write a lot of code. And then this code will be automatically run in a sandbox with the result. And then you will just keep doing it. And yeah, just going through all these iterations. And it generates some visualization as well, like the spatial organizations. Yeah, and then you will, once it gets it done, you will also like flag this step is done and then just continue the next step and continue until all the steps in the plan are finished. And you will return to the user with the final report. Yeah, that you can immediately use as well. Cool. Yeah, and it's available right now at biomni.stanford.edu. Feel free to try it out as well. And I think since we launched it, we're also able to amazingly find out a lot of drug discovery people, like real world, it's like moving beyond academia, right? So this is a number of Biomni users in the first 80 days. Also able to amazingly find all these pharma scientists actually using it across different use cases, ranging from target discovery, like ad meta property predictions, and all the way even to the clinical side of things. Cool. So I think that's was the first project, the first phase of the Biomni project. So what's next? I will share some more recent works on kind of things that we are very excited about. So I think it briefly kind of it generally falls into two categories. The first one is want to achieve expert level performance, and the second one is this kind of when we have the generalist biomedical agent, the foundation layer, then we can start building the agentic system to compose them and to get even more kind of new capabilities. So here I will demonstrate hypothesis validation and also to develop some biology, new algorithms as well. So the first task is how do we achieve expert level performance? Because right now Biomni can get a task done and give you a report no matter what task you give. But it's also, yeah, we're able to see that for many tasks, it's able to give like expert level performance. But for also a long tail of many tasks, it's not there yet. So how do we improve the agent in a systematic way to achieve expert level performance for any tasks? That's kind of a very important problem. And I was thinking if there's a scalable way to do it as well. So as Hanchen mentioned, like reinforcement learning recently has got a lot of attention because it's a extremely scalable way. You just define the data, the reward, and you just train the agent. It will figure out on its own. Like heel climb on its own, right? So the idea is very appealing, also for biomedicine as well, because we can collect a lot of biology data with some reward, and then you just train the LLM end to end, and then just improve it. And actually, the RL is also, I think, very interestingly how is a kind of enable potentially a new way of doing things, right? Because if you think about how our current way of doing things, it's also scientists follow some like a protocol, some convention, like sequence of steps. And this convention was also generated through these trials and errors by a group of researchers maybe like 10 years ago. But it's not necessarily the optimal way because it could be this more optimal sequence of steps to it. And I think the RL is a very nice way because it basically asks it to do massive number of trials and errors. And then given a reward, it's able to say, okay, this path is actually more optimal. And then maybe it's it could be a way to learn this kind of a superhuman level convention, a new way of doing the same task. So we're inspired by that, and we built the first version of it, which is called Biomni R0, because we think there's so much things to add on, so that's why we call it R0 instead of R1. But the basic idea is like we still use our environment, the E1, and also the A1, which is the agent architecture. It's like a very simple agent architecture, and then we do like trajectory, like rollout. So you collect a bunch of trajectory with the answer from the agent. And if we define 10 tasks with a predefined reward. And you can let the LLM know, okay, for this kind of trajectory, you're doing it really well. And for this kind of trajectory, it's not. And then the LLM, during the backpropagation, is able to automatically figure out what kind of steps is important, what kind of step is not. And then by aggregation, it's able to generate a new trajectory that is kind of optimize the reward. So this joint work done with led by Ryan, and also collaboration with Shi, Yuanhao, Yuri, and also with the Sky IR group at Berkeley. And for some initial results, we're able to see that even use some open source models, like 8 billion queen models, after training, it's able to get really good performance on in-domain tasks. For example, here we are able to have 85% improvement, and actually the performance is actually on par with the closed source models as well. And if we use a 32 billion model, it's actually even able to outperform this closed source models. And because the performance is so significant, we're thinking this is potentially a very interesting recipe to kind of do the hill climb and achieve expert level performance. And we're also able to find that improvement is also very consistent across different subtasks. So here, this is the list of 10 tasks we are using for now. CRISPR delivery, GWAS causal gene prioritizations, and also patient gene detection. It's also like designing the screen and things like that. And you can see that actually this agent, I think in this 10 tasks, I think our agent has the best performance in like 7 to 8 tasks as well. So it's also very consistent. Yeah, so I think after that, we're also thinking a lot about, is this really the way to go? We're still thinking about it. So we'll be also curious to hear your thoughts as well. But if we really look at, like, first of all, is it like similar to humans? Because for IL, it actually requires hundreds of examples for the same task, for the trials and errors as well, but it's roll out. But for real world biologists, they don't have the privilege to do hundreds of trials, right? It's so expensive to run on one task, and they just need a few. And also the biology domain, like I think one of the hardest challenges that we face when we do the R0 project, actually the reward function is very hard to define. There's many other tasks, there's just no reward. So because it just biology is intrinsically low data, right? So how do you kind of define rewards in this setting in a large scale fashion is also a bit unclear. And recently, I think there's also lots of discussion around this idea around system prompt learning, where you just try it once, and you have another LLM to reason, okay, what kind of lessons did you learn? Then put the lessons into the prompt as kind of in the memory or something like that. So we're also exploring all these kind of ideas, trying to achieve expert level performance in a more kind of data efficient fashion. So that's the first category of tasks we're looking at right now. And another task is also another category is like this multi-agent systems. So as Hanchen briefly mentioned, I think getting a task done is just one component of this more global hypothesis validation. So typically, given hypothesis, there's usually multiple different kind of falsification experiment that you want to do to really falsify a hypothesis. So here, in working with Ying, Ryan, Michael, and Emmanuel, and Yuri, we proposed a new method that actually it was based on Biomni agent that interleave multiple of them, and then to have a more rigorous approach to do hypothesis validations. I think the idea is like we have one agent, which is called the experiment design agent. Given the hypothesis, it will basically, we ask it to do some iterations to design a falsification experiment to falsify the main hypothesis. And then after it is done, we'll have an experiment execution agent, which is based on Biomni to execute it. For example, like writing a lot of code, observe it, and then the result, and then just continue and generate p-values. And then because usually for a main biology hypothesis, there's often usually all kinds of different falsification experiments. We need some way to aggregate it in a rigorous fashion to have some kind of sequential error control. This relates to the e-values that Hanchen mentioned to give like more rigorous error control in a sequential fashion. And so overall, we're able to, this agentic system is able to kind of control the false discovery rate, control the type 1 error rate, and then still improve power by a lot. So we tested out in the real world, we recruit like 10 PhD level scientists, and then give them the same kind of hypothesis to validate. So they and we're able to find that they have similar type-one error rate, similar power as well, and also it takes much shorter for Popper, but takes it generates much more experiment, writes more code than humans. But interestingly, it's able to also find kind of they're doing pretty similar things, right? They're designing similar kinds of falsification experiments, like testing the expression correlation from GTACs, interaction network from some physical from protein-protein interaction networks, EQTL data sets. And there's also proposed some new ones, and human also has some new ones as well. And also they're using all kinds of different diverse statistical tests as well. So we're able to see that like by composing this agent together, we're able to do like more high-level tasks that can enable like new capabilities for scientists. And lastly, I also want to highlight a new work that's in collaboration, which is led by Alistair, working with Lei and Martin here. It's basically using agent to also develop bio algorithms. So actually, the first few years of my PhD, before the agent stuff, I was also just developing these AI bio models, like just on the biological data sets. And I often find that the best model is often involves by embedding this kind of biological inductive bias right into the model. So you need to know both AI and biology deeply in order to create like a customized model, like AlphaFold. AlphaFold is a great example, like it embeds all this kind of prior knowledges, the deep knowledge about the data, and convert it into the modules in the AI models and then make it really work. So we think the AI agent is actually maybe for the first time maybe the best single entity that knows both AI and biology so deeply, so it can potentially develop much better AI plus bio models. Yeah. So here we created the first versions of it, like an initial attempt, which the basic idea is like given a task, you have some kind of the validation set as well, and then you ask the agent to collect all this domain knowledges and then propose some solutions, and then start iterating on these solutions. And for this, during the iteration for each step, you will have the validation set performance. And then also from the literature, we also gather a lot of instructions on how do you update these solutions. And there's a lot of details as well, but overall, that's the idea. So you can iteratively just optimize this algorithm in order to beat, and in order to have like great validation set performance as well. So in the paper, there's much more in-depth benchmarks, but I think really interestingly, we kind of Alistair really apply it to human genetics tasks. For example, this is SCDRS, which was proposed by Martin while he was actually had here. So it's like a disease-critical cell type detection algorithms that has like kind of full secondary control while I need to detect as much disease-critical cell types as possible. So the Tussle AI just takes in the original SCDRS and then just start optimizing it. So also notably, the Tussle AI can do both optimization from existing algorithms, also just completely generate from scratch as well. But for this example, it's basically generating from some base checkpoints. But it's able to have like much better power, like 40% more power. And then it's able to also have kind of same level of force discovery rate control as well. And then it's also able to detect like novel cell types that match with the ground truth, and also detect novel cell type when we apply it to some autoimmune diseases. And interestingly, it takes 24 hours. And the agent tried like 136 versions at a cost of 30 cents. But the original author took like 10 versions of it over 3 months to get it done. So we're really thinking in the future, maybe the next generation of AI plus bio models actually generated by agents. So we need to actually train the agent to how do we generate these AI plus bio models. Cool. So I guess that's all for my talk. Thank you. I want to thank my co-author again. And this is also my contact information.